We are going to now talk about JAK-STAT signaling system. It is a very important signaling system. It is utilized by signaling molecules that defend us against intracellular parasites. For example, viral infections. Gamma interferon, when it is released in the body in response to parasites invasion of the host body, it increases antigen presentation of macrophages. I know we have not talked about immunology yet, but basically what this means is in response to gamma interferon, cells, specialized cells, can present pieces of virus to the immune cells, activating them and telling them that there is a viral infection. It activates the lysosome activity. We know lysosome is an organelle in the cell that can digest ingested material which cells have ingested. So lysosome activity of macrophages is enhanced. It also causes expression of special proteins that can grab viral particles and display it on their surface. So cells are now marked and telling the immune system that they have been infected by an intracellular parasite. It also promotes adhesion and binding required for leukocytes or white blood cells which defend us against pathogens. Basically, the gamma interferon is a molecule that is signaling the immune system that there has been an intracellular parasite, a parasite that is living or hiding inside the cells. So the whole immune machinery focuses on looking for this parasite and finding the cells that are infected with this parasite and destroying those cells. We will talk about this process in more detail when we study immunology. But this is a very important molecule that helps us defend against viral pathogens. So how does this molecule work? Let's look at that. The receptor, interferon receptor, when it binds the ligand, it dimerizes. This receptor does not have kinase activity or enzymatic activity on its own, but it is attached to proteins that have enzymatic activity. Here, for example, JAK1 and TYK2, these are the two proteins that are attached to interferon receptors. Binding brings two JAKs close enough to cross-phosphorylate each other, increasing their kinase activity. Previously, we saw that when two receptors came together, they cross-phosphorylated each other and their kinase domains became very active. Here, it is the same principle, but the protein, the enzymatic activity is present on a different protein. When JAKs are phosphorylated, they phosphorylate specific regions of the receptor, and these phosphorylated amino acids basically serve as docking sites for another type of protein called STATs. So again, when we talk about different molecules, it is not that there's only one type of molecule. There are several different types of STATs, several different types of JAKs. We'll talk about them. So here, the phosphorylated receptor is basically serving as a docking site for STAT proteins. These STAT proteins have a special domain, SH2 domain. We have talked about pH domain. SH2 domain is basically domain where there is a phosphorylated tyrosine residue. So these domains recognize that structure and bind to it. This brings STATs in close vicinity of JAKs or TYK2 in this case. They also phosphorylate STATs at special residues, again generating tyrosine, phosphorylated tyrosine residues. Now imagine that this is also SH2 domain basically of the STAT molecule, basically meaning that it can bind a phosphorylated tyrosine. Here we also have a phosphorylated tyrosine. So these molecules, when they're phosphorylated by JAKs, STAT molecules, when they're phosphorylated, they dissociate from the receptor and they dimerize. Since both of them have SH2 domain and they have a domain to which SH2 domain can bind. So this results in release of the STAT molecules and their dimerization through the SH2 domain, and this dimer can actually now go to the cytoplasm and go to the nucleus directly, and in combination with other gene regulatory proteins, it can bind to a specific DNA segment response element and stimulate transcription of specific genes. So here we have seen that this is a very shortcut, one of the direct routes from the cell surface directly to the nucleus. So here you can see the whole scheme: receptor dimerizing. Receptor is already attached to JAKs. JAKs cross-phosphorylate each other, which increases their kinase activity. They phosphorylate the receptor. The receptor, when it is phosphorylated, recruits STATs. STATs get phosphorylated. This causes a conformational change in STATs. STATs are released. STATs dimerize. They go to the nucleus and they recruit other proteins, gene regulatory proteins, resulting in transcription. So this is one of the directest routes from the cell surface to the nucleus. I'll talk about some of the JAK-STAT signaling proteins. These are, for example, erythropoietin. It stimulates erythrocyte precursor cells to survive, proliferate, and differentiate. And it is associated with JAK2. Erythropoietin basically stimulates the cells that are going to make red blood cells. So it is also a protein. It's also given to people who have difficulty in making red blood cells clinically. JAK2 deficient mice, they have developmental abnormalities and they die very early in development. So JAK2 is very important because mammals, they cannot survive without red blood cells. Prolactin is another signaling molecule that stimulates JAK-STAT system. It results in production of milk. It activates STAT5 molecules. STAT5 molecules stimulate the transcription of genes that encode milk proteins, for example. Alpha interferon, we talked about gamma interferon. It activates both STAT1 and STAT2. However, gamma interferon just activates STAT1 molecule. So here is an example how the signaling molecules that activate different JAKs and different STATs is present on the screen, is presented on the screen. You can look at that. We have already talked about some of them. So I wanted to give you an example of a system that can detect a signal and convey the message directly to the nucleus to produce special type of proteins. We'll look at other systems that are also directly wired to the nucleus. There's a direct wire from the cell surface to the nucleus in the next module.